children, in the previous topic, we discussed variation and heredity. Now you know that variation is the difference in the characteristics of offspring compared to their parents. Heredity is the inheritance of parental characters by their offspring. You also know that pioneering experiments in the field of genetics were conducted by Gregor Johann Mendel. And he is known as the father of modern genetics. I am Shane, will now explain the very important and interesting topic of Mendel's experiments and laws from the lesson Heredity and Evolution of Class 10 Biology. Mendel, 1822 to 1884, was an Austrian monk who discovered the basic principles of heredity. Through his experiments, which are the foundation for modern genetics. He was born to a family of farmers in 1822 and later joined the Augustinian monastery at Brunn. Mendel went to the University of Vienna for training and returned to the monastery in 1854. He worked as a priest and teacher in high school. In his leisure time, he started his famous experiments on the garden pea plants. Learning outcomes. After studying this topic, you will be know about Mendelian laws, differentiate between phenotype and genotype. Understand the process of monohybrid and dihybrid. Cross experiments. Keywords: traits, phenotype, genotype, heterozygous, homozygous, independent assortment, allele, monohybrid cross, dihybrid cross, let us see the concept map on heredity. Mendel worked with nearly 10,000 pea plants of 34 different varieties which differ from one another in many ways. Thus, Mendel had chosen 7 pairs of contrasting characters for his study as shown in the table. Mendel's experiments. Due to the following advantages, Mendel selected garden peas, Pisum sativum, plants for his experiments. They can be grown easily in conducting experiments on open ground or even in pots. They have a short growth period and life cycle. They give self-pollinating flowers and easy to conduct cross-pollination artificially. They produce a large number of seeds. They show contrasting heritable characters. They produce fertile hybrids through cross-pollination. Mendel performed experiments by crossing the pea plants. He observed that the inheritance of contrasting forms of a single character can be studied one at a time. For example, tall and dwarf forms of a single character contribute to the height of the plant. Similarly, the grey and white forms are a single character of the color of the seed coat. He then obtained pure or true breeding varieties of each of the contrasting forms by allowing them to self-pollinate for several generations. By this, he got pure tall offspring and pure dwarf plants. Cross-pollinating plants pure breed of yellow and green seeds gave the first filial or F1 generation or progeny of first generation parents producing all yellow seeds. These F1 pea plants on self-pollination gave a second filial or F2 generation resulting in about 75% yellow and about 25% green pea plants. Third filial or F3 generation resulted in about 25% only yellow seed giving pea plants. The rest of the yellow seed giving pea plants gave about 75% yellow and about 25% green seed giving pea plants. Set of green peas gave only green seed giving pea plants. Mendel made some assumptions by which he could explain his observations. In the F1 generation, we can observe that 75% are yellow seed producing plants and 25% are green ones. This is known as phenotype or externally visible character, and this 3:1 ratio is called phenotypic ratio. Genetically, in 75% of yellow seed producing pea plants, only 25% of pure breed pea plants produce yellow seeds. These are homozygous and have the same factors for representing a character capital Y, capital Y. The remaining 50% yellow seed producing plants are heterozygous, capital Y, small y. The remaining 25% are pure green seed producing plants or homozygous type small y small y. The seed color of plants is represented by letters capital Y and small y. For yellow and green respectively are known as genotype, and the genotypic ratio is 1:2:1. A cross between two plants differing in one specific character like tall and dwarf or yellow and green seed color is called a monohybrid cross. The monohybrid cross ratio is 3:1. The cross between the tall and dwarf plants resulted in all tall plants in the F1 generation. But they must have contained the factors for both tallness and dwarfness. This is because when F1 plants were self-pollinated, the F2 generation produced some dwarf plants along with tall plants. The factor for dwarfness, however, remained hidden in the F1 generation. Mendel called this factor for tallness dominant over dwarfness, the recessive factor. The F1 plant is heterozygous and inherited a factor for tallness from its tall parent and a factor for dwarfness from its dwarf parent. Dihybrid cross involves the inheritance of two pairs of contrasting traits at the same time. The two pairs of contrasting characteristics chosen by Mendel were the shape and color of the seeds, like round yellow seeds and wrinkled green seeds. Mendel first crossed pure breeding plants having round yellow seeds with pure breeding plants having wrinkled green seeds. Only round yellow seeds were produced in the F1 generation. From this, it was concluded that the round and yellow seeds were dominant over the wrinkled and green seeds. When the hybrids of F1 generation pea plants having round yellow seeds were crossed by self-pollination, then 4 types of seeds with different combinations of shape and color were obtained in the F2 generation. A dihybrid cross produced 4 types of F2 offspring. In this, 9 plants with 2 dominant traits, 3 with 1 dominant trait and 1 recessive trait, 3 with another dominant trait and another recessive trait, and 1 with 2 recessive traits. 2 new combinations of traits with the round green and wrinkled yellow appeared in the F2 generation during the dihybrid cross. The ratio of each phenotype of seeds in the F2 generation is 9:3:3:1. It is known as the dihybrid ratio. The factors for each trait remain independent and maintain their identity in the gametes. Punnett square is a checkerboard devised by a British geneticist, R.C. Punnett, for the study of genetics. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. Based on the experiments of monohybrid and dihybrid cross, Mendel proposed three important laws called Mendel's laws of heredity. Law of dominance. When two homozygous individuals with one or more sets of contrasting characters are crossed, the characters that appear in the F1 hybrid are dominant, and those that do not appear in F1 are recessive characters. Law of segregation or law of purity of gametes. When a pair of contrasting factors are brought together in an F1 hybrid, the two factors of the allelic pair remain together without mixing and separated during gamete formation, so that only one factor enters each gamete. Law of independent assortment. In case of inheritance of two or more pairs of characters, simultaneously the factors or genes of one pair are sought out independently of the other pair. This is all about Mendel's laws, and in the next topic you will learn about evolution. Stay tuned.